All right, hey guys, happy Tuesday. Hopefully Monday went okay for you. A little cold today, a little breezy, a little snow yesterday, but yesterday you guys were taking a little quiz on kind of antibiotic resistance, and it kind of dealt with what was talked about maybe before the weekend with MRSA staph, which was the reading on Thursday, I think, and also just on antibiotic resistance and the videos that were on the previous day, Wednesday. So some people did really well, and then some people not so much. And then just in my video, I said, hey, make sure that you're reviewing that stuff. And if you hadn't done it, to go do that. I'm guessing some people didn't because the questions were honestly, a number of them were straight from that reading. So anyway, you should have did okay if you did the work. At least okay. So I'm going to talk a little bit about how they develop resistance and really deals with how they reproduce. So bacteria can reproduce very quickly, and because of that, the generations of bacteria can accumulate very quickly. So like a human generation, we can kind of estimate to be about 25, 30 years or something like that. Where in bacteria, bacterial generation can be about on average about 30 minutes, but it can be about as quick as 20 minutes. So how do they have such quick generations? Well, they really are just one cell big, and they just needed to make a new cell, a new version of themselves. So I'm going to explain this diagram to you, and then I'm going to have you guys write a few sentences in your notes. So you'll need to have those open. Here's our cell, and that cell is got some DNA in it, and we call that loop of DNA in the bacterial cell a chromosome. And what has to happen before the cell replicates is it replicates its DNA. This may sound familiar. It's pretty similar to the cell cycle or mitosis, but it has some differences. So once that DNA is replicated, the chromosome will kind of split apart and the cell will split down the middle. And a lot of times it's got to split the cell membrane and a cell wall. And this results in two identical cells. So this is what's known as asexual reproduction. That is reproduction without a mate. So the cell is just picking a time and dividing into two new identical cells, and they are exactly the same as the first one, this except for size. They will be smaller in size. So that's basically what's called binary fission. Fission means to split, bi meaning two, so we're splitting a cell into two. So explain this diagram using a couple of sentences is your job. So you can go in this box and you can write in your own explanation. If you want to pause the video now, you can. I would say something like, hey, the cell multiplies its DNA, and then it splits down the middle, and a copy of each DNA gets put into each new cell, and this takes as little as 20 minutes, something like that. So go ahead and write yours in now. So now that you guys are done with this box, I will say I added a little video here, and I'm going to post that video onto Google Classroom. So the link will be on Classroom, and you'll be able to watch it. It'll be right underneath this notes. So go ahead and watch that afterwards on binary fission, because we will come back to it. Okay, I'm going on to the next slide. And this is, it's definitely going to be different than binary fission. This is when bacteria decide that things are not going well, okay? So bacteria can then sexually reproduce, and that is finds a mate and swap some DNA. That process is called conjunction. You'll see that word right here, and it's a form of sexual reproduction, okay? So basically what's going to happen is once conditions are not good, you're exposing bacteria to an antibiotic, or your body develops a fever and it's too hot for the bacteria to really survive, there are loops of little segments of DNA that are not the chromosomal loop in here. We call it plasmids, and maybe you saw that in that other video if you already watched it here or on Google Classroom. Those plasmids can be spun off of the main chromosome. It's just a piece of DNA, and it's copied, and they can move a copy over to another cell, effectively giving another cell that piece of DNA. This has the same effect as swapping some DNA in sexual reproduction or recombining genes. in different combinations. And that means that the bacteria that result, the two cells after a conjunction event are different than the two cells that met before the conjunction event. So I would advise you guys to go ahead and click the play button on your notes here and watch this video on conjunction. And again, the reason is that is diversity. We're changing up what the bacteria have as far as DNA. We're adding diversity. We're shuffling genes, and because of that, you have made bacteria that are different than the parent bacteria. And when that happens, different bacteria may have a better or a worse chance of surviving an event that is difficult for them. So it could be a drug like an antibiotic that's killing everything off. If this one bacteria has a piece of DNA that makes them stronger, they can then give a copy of that to another bacteria and therefore make that one stronger as well. Anytime you have more diversity, we have more of an ability to survive things like, well, COVID virus. It seems that some people are affected really heavily by it, and then other people who have different DNA seem to, well, some people can walk around with the virus in them and not really show any symptoms. And we're looking into this a little bit more, but there are people that can carry the virus and not get sick right now. That's what we found. So the diversity in our DNA allows us to have these kind of immunities or different responses to difficulties in our daily lives. So I would like you guys to make sure that you do slide 32. Thanks for watching the notes. I'll link this video on Classroom as well, so please watch that. And then I would like you also to watch this video on conjunction on slide 33. Thanks for watching this one too. It really helps, I think, if you're, for as far as like watching instructions and stuff go, so that those of you that do watch it kind of know what to do. So